Finish up this section. So where we left off was talking about nucleotide biosynthesis. We talked about amino acid biosynthesis and in some of the initial pathways for other biomolecules, but let's do some nucleotides too. All right. So there's really two main ways that we make them, and that corresponds to our purines and our pyrimidines. Now, of course, we can scavenge nucleotides that we consume, but we're talking about de novo, just kind of making them in the body. And we can make these from amino acids and ribose 5-phosphate and carbon dioxide, and of course, our old friend ammonia that is so important to maintain a balance of. So we can salvage them and we can make them from initial. Interestingly, there are a lot of parasites in there, like malaria, that lack the de novo biosynthesis pathway and rely exclusively on salvage. So if you can find various drugs that will inhibit salvage pathways, that's one of the ways to treat malaria. So you get some anti-parasite drugs in there. Of course, that's a little harder said than done, but anyway, oops, come on, there we go. All right. So one of the good things about this, pretty much the biosynthesis of nucleotides is really similar, if not identical, across all organisms. So they all do it the same way. The bases are synthesized while they're attached to ribose. So we have our ribose in there, and then we build our base on there. Glutamate provides most of the amino groups. Glycine is the precursor for purines, and aspartate is the precursor for pyrimidines. Nucleotide pools are intentionally kept low, so cells have to continually synthesize them. This may actually end up being a form of regulatory control because you can limit the rates of transcription and replication because you have to make nucleotides to do that. Otherwise, so you don't get this cells that are rapidly growing and dividing and so on in there, or transcription just happening over and over again. All right, so let's talk about purines first. If this were a graduate level, oh my God, memorize all this. Where does this carbon come from? Where does this carbon come from here? But here we have our purine. We can see we have glycine, and we're going to get a carbon from a couple carbons from chromate, a carbon from carbon dioxide. Yeah. No. What you do need to know is that for purines, you start with glycine and then you'll build. And you see a nitrogen from aspartate, a couple nitrogens from glutamine in here. But let's talk about how this happens. Our biosynthesis of purines starts with our PRPP, phosphoribosylpyrophosphate. If you remember that from a couple chapters ago. And when we'll see adenine and guanine, they're synthesized in their monophosphate form. Okay, so we start with PRPP. And then the purine ring will build up and following the addition of 3 carbons from glycine. Okay, so our first intermediate with a full purine ring is this inosinate. Okay, so we're in inosinate monophosphate to be particular, and it's monophosphate for this group over here. Okay, so here we have our phosphoribosylpyrophosphate. We start adding stuff in here. Here's our glycine that's going to come in and attach to start building our ring structure. And then we start putting the rest of the stuff on here, and we have multiple steps where we stepwise add 1 or 2 atoms at a time, and ultimately we end up with air or aminoimidazole ribonucleotide. Okay, so we have something like this. So here's part of our ring structure. Okay, and you can see we've added a carbon over here and a nitrogen over here through this. And then we continue this process and start building in the other ring for our purine. So remember, our purines have that double ring system. We keep going. We go from air to care, well, N5-care to care, and so on, and then psych-care, and then sarcasm. No, sorry. We just keep on going, keep adding stuff, and close that ring. And so now we have built this ring on our inosinate. Okay, so here's our purine attached to our ribose that is monophosphorylated. So inosinate monophosphate. Okay, 11 steps just to get to here. Okay, now this still isn't adenine or guanine, so we got to make a few changes to do that. But our inosinate monophosphate, or our little imp there, we can use aspartate to get it to get rid of this oxygen and add an amino group there across a couple steps, and that gives us our adenylate. I remember this aspartate adenylate AA. Okay, if we have this imp dehydrogenase, okay, then we get this xanilate monophosphate, and then we can replace the oxygen that we've added to the ring structure with a amido group. So we have that aminotransferase, and we get guanylate in there. So imps are the original OGs. I don't know for guanylate, but basically for purines, you have this big, long 11-step pathway that gets you to an inosinate monophosphate, and then you can branch off to get either adenine or guanine. Okay. All right, and that's the only thing I want you to take away from that. Big, huge step. You start with the ring structure. You start with glycine to get those 3 carbons on there, and then you build, connect rings. You get IMP, and then you can convert that either into adenylate or guanylate. Okay. Not too bad, right? Okay. Yeah. All right. How do we regulate this? Oh, there are 4 major mechanisms of regulation and lots of minor ones in here. So our glutamine PRPP aminotransferase is inhibited by the end products. So not only our AMP and our GMP, but even our IMP can feed back and inhibit that early step in there. You can also have excess GMP, which will inhibit the formation of xanthylate. So for the particular branch there, if we have lots of GMP, it'll feed back and then shunt this over to the AMP pathway. The same way AMP will do the same thing. If we have excess AMP, it'll feed back and shunt stuff over into GMP. Okay, pathway in there. Both of those, if they have high concentrations, can go back and inhibit the phosphorylation steps. And then once we add another phosphate group to AMP and GMP, so we have ADP and GDP, they can go back and inhibit the very first enzyme in the pathway. All right. So again, none of this you need to memorize, but you should be familiar with this. You may get a question like, suppose there's a drug developed that binds to, and I might pick something in one of these intermediate steps here, and you have to go back and kind of recognize, find that step and say, oh, what's this going to do to concentrations of AMP, or maybe I'll say CMP or something like that. All right. So just kind of being able to kind of suss this out, but these are our major regulatory steps in here. All right. And of course, I get asked a question like, I know I had a similar question on the last exam about there's a mutation in the binding site that won't allow IMP to bind. How would that affect the formation of IMP? So you probably make more of it because you're not going to get feedback inhibition, but it's not going to be a massive accumulation because your GMP and AMP concentrations can inhibit it as well too. Of course, it could be, it could all go haywire. So there's a lot of possible right answers, and it just comes down to the reasoning, which is what I'll be looking at. Okay, does that all make sense? Okay, all right. So that's purines. That's one way. So we got our big ring and one way of making those. Our pyrimidines are a little bit different because our purines, we have our base there, our ribose group, and then we have a little bridge there, and we start building the scaffold up there and make it. For pyrimidines, we make it first, and then we attach it to our base, to our ribose. Okay, so we make the so we make the pyrimidine first in this oro-rotate. Okay, and then we attach it to ribose 5-phosphate, and so we kind of do it in the opposite direction. All right. Aspartate and carbamoyl phosphate. Where does carbamoyl phosphate? Where do we find that? Think of R. Kelly. Yeah, urea cycle. There we go. Terrible, right? All right. Okay. It's the R. Kelly mechanism. Okay. Yeah. So anything that helps you remember it. So aspartate and then carbamoyl phosphate are going to come together to give us our initial little structure in here. And then of course, just like we saw 11 steps to get to IMP, we're going to have a whole bunch of steps to get to this oro-rotate. Okay. So, so we had, Ororotate. Once we have the ororotate, there's a ororotate phosphoribosal transferase that will attach it to our ribose 5-phosphate. And then once the ororotate is on there, we will further modify the ororotate into either cytidine or thymidine or uridyl. Actually, do uridyl first. Okay. Okay. Okay, so UMP gets phosphorylated to UTP, and then we can convert UTP to CTP, and I think TTP should be in there somewhere as well too, right? Yeah, all right. Okay. How do we regulate this? All right, again, feedback inhibition, end product CTP, and it's accelerated by ATP. So if we have lots of ATP, we're gonna say, yeah, we don't need ATP or GTP, make more CTP and UTP, TTP, depending on what we have there. So we can see our normal activity. We're not gonna make, with no CTP there. If we have lots of CTP, then this enzyme slows down as it feeds back. So this is just a little diagram here saying in the presence of CTP, our velocity of reaction is significantly slower, like half the speed than if we don't have any CTP present. Okay, so feedback inhibition. All right. So, we talk about UMP and UTP and CTP here. What about TTP? Our ribonucleotides are the precursors to the deoxyribonucleotides. So these are catalyzed by the conveniently named ribonucleotide reductase. Deoxy, we're going to reduce them. So 2 protons get donated by NADPH, and they're carried by these big pathway in here. I'm not going to focus on this because I know when you start seeing these pathways, you're what? This is a big chain of redox reactions. We've seen a couple of these before, but the key thing is our ribonucleotide reductase is going to be involved in electron transfer to remove that oxygen. Big structure in here where we can fit our stuff in and we can have our stuff going on in there. I'm not going to ask too much about that. I'm not going to ask about the mechanism, but it is proposed to be a free radical mechanism. So again, radical chemistry is really powerful chemistry. It just has to be well controlled. If you think about nuclear energy, most people, when they think about atomic energy, atomic bombs, uncontrolled, boom, it all explodes in there too. That'd be really bad for the body. Nuclear power actually produces probably the most power. It's aside from the cost of regulatory and containment and all that stuff in there, even factoring that in, nuclear energy is the cheapest energy out there. So our radical chemistry is very powerful. It just has to be well controlled in there. So that's why we see all that glutathione reductase, where is it in here? Yeah, glutaredoxin, all these things in here that are helping make sure our single electron transfers are going where we want them to go. Anyway, the whole key in that is that we want to convert and reduce our ribonucleotides to deoxyribonucleotides. We want to get rid of that 2' hydroxyl group in there. Okay. You may see a question about ribonucleotide reductase regulation, because everything's starting in this chapter, everything is going to be focused on regulation. So there's 2 regulatory sites. You normally see something bind here and affect it. Well, we got 2 spots to affect it. One is going to affect the activity. Okay, so if we have lots of ATP, it will bind and affect it. If we have lots of deoxy ATP, it will bind and inhibit it. Okay, that's pretty normal. The other regulatory site is going to affect the substrate specificity. All right, so remember, we got our 4 different nucleotides, whether they're deoxy or regular ribonucleotides. So if one of those is too high, it can bind to a site and say, don't. If we have lots of CTP, it's like, don't bind so much CTP, bind these others. Take like the old vaudeville comedians, like, take my wife, please. So what we see here is, in this case that we have on the slide, we have lots of ATP or DATP, it will bind at this other site and make the enzyme less likely to bind ATP and convert it into deoxy ATP. it will enhance the specificity for UDP and CDP, and so on and so forth. So each enzyme can bind there and change the specificity for the others. And one of the ways this happens is through oligomerization, where we can see these all kind of bind together. Now, I don't know if you guys like this image. Sometimes this makes sense to me, sometimes it doesn't, but this is kind of like, oh, we have lots of ATP, we'll bind here and say make more of this instead. So if it makes sense, use it. If it doesn't, you can ignore this. The words will work just as fine. Okay. All right. So anyway, thymidylate, how do we get that? Okay, because we start off, for our pyrimidine synthesis making UTP and CTP, all right? So there's a little bit of a roundabout pathway. We make our deoxy-UTP first, and then we're going to dephosphorylate that into deoxymonophosphate, and then we will convert the uridylate into a thymidylate in there through this thymidylate synthase, which basically brings in a methyl group from tetrahydrofolate. Remember our THF, a good single methyl group carrier in there. It's really kind of neat because thymidylate synthase is a target for some anti-cancer drugs. Why is that? What do we know about cancer cells? Yeah, and they grow real fast. So we have this rapid division and so on. So if we target thymidylate synthase, then they can't incorporate thymidylate in there. And this is why cancer patients often have upset stomachs and the hair falls out, because we can't target it. Well, we're getting better, but in general, early cancer drugs, you could only target fast growing cells like the epithelial cells of the stomach, the hair cells, and so on and so forth. So that's why, cancer patients who are undergoing chemotherapy would often lose their hair and have seriously upset stomachs. This is also why you cannot give these drugs to people who are pregnant, because fetuses are fast growing cells as well too. But cancer cells, fast growing cells. So you target this and that helps kill the cancer cells. Okay. So again, if we look at our biosynthesis of thymidylate, we start off with either CDP or UDP. We put that in the deoxy form. Then we add a kinase in there. So we make our triphosphate section. Then we'll cleave off pyrophosphate, and then thymidylate phosphate will add a methyl group to dump to make our D-tump in there, whatever. So, and here's our tetrahydrofolate adding our group. Yeah, I want to see the actual picture of it. Well, anyway, don't have the picture of it there. All right. There's another issue. If you are pregnant and go visit your OBGYN, one of the first things they're going to do is hand you a bottle of vitamins, which will include folic acid. So folic acid deficiency, very widespread, especially if, so I always think when I was finishing up undergrad, and my one roommate moved out, so I got a replacement roommate that he found, and it was a girl who was pregnant, and she was fairly well along, but pregnancy cravings apparently are real things. She ate a lot of boiled sausages and potatoes, and maybe not a lot of vegetables. And I was that later on when I went to school, I was like, were you getting enough greens in there? Like almost raw steak. Yeah, Pittsburgh style. It's like it was warm on the outside. I could never do that. Yeah, I've moved up. Yeah. But folic acid, which you find often in leafy green vegetables, is critical for neural crest development of a fetus in there. So if you do not get enough folic acid, there is a strong chance of some type of birth defect in there, and you can have kid born at best with some cognitive issues, because the brain just isn't well formed in there. So that's why, quick and easy way, take vitamin B12, our folic acid supplements in there. Just take one of these vitamins every day, and that provides enough folic acid to make sure that neural crest tube, whichever it is, the formation will occur, and you'll get some good development in the fetus in there.
So is it
Um, yes, yeah. So reduced thiamine causes uracil. What will happen here is if you don't make enough, you can incorporate uracil in there. The repair mechanisms to get rid of uracil create strand breaks. So you actually have to cleave the DNA and then replace it with something else. But strand breaks are bad. They're hard. You can see this with like was it ionizing radiation. One of the reasons why radiation poisoning is so bad is because, again, repair mechanisms, you have to cleave. Not only do they create dimerization, but you also have to, in order to repair it, you need to break the DNA and put in something new to fix it. So when you have a lot of damage and these repair mechanisms are firing up, there's a chance that the repair isn't very good. If you think about like maybe repairs that have been done in your home or something like that, you look at the quality of repairs and it depends on. I remember when we first moved into our house, we had one of those home warranties. And here's what I figured out about those home warranties: they're peace of mind for new homeowners. But who works for the home warranty? People who can't compete in the actual marketplace. It's like we never had, and unless it was a really, really simple job. Because I know we kept our home warranty for like 2 or 3 years, and then finally I was like, why? It's like every single repair company in there was not good. They could handle something if it was really simple, but these contracts kind of kept them in business because word of mouth or the work that the quality work they did was not sufficient to garner them enough business. They didn't do quality work. So it's not the best analogy, but if you have lots of uridylate incorporated in there, and you constantly have to have those repair mechanisms in, it's going to repair the DNA, but it's going to make mistakes, and that error rate of the repair, neurological impairment is the one that you'd see most commonly with fetus development. But even for older people, if you don't get your vitamin B12, you can promote cancer and heart disease that way. So eat your greens. I don't know if your parents say. It's funny. It's like so trying to change my diet and be healthier. It's like I was eating raw broccoli the other day.
Just by itself?
I think I put like a little bit of dip on there. Yeah. But ranch is in. But yeah. Yeah. I was not a big fan of ranch, but I just ate a little bit raw. We were at a party there too. I was like, I can't eat just all the meat that's here. So they had Brussels sprouts as well too. Yeah. And it was.
I like my boiled. I don't like my.
Oh yeah, I like them roasted. I like them crispy. Yeah. But it's that's why you got to eat your vegetables. So, and I'm so lucky that my daughter loves vegetables. She keeps saying she's a vegetarian, but no, she just likes to eat lots of vegetables.
So those that's found commonly in.
I think leafy green vegetables. So like kale or spinach green, spinach. Yeah. So yeah. I try and eat more salads. It's funny because I don't really consider myself a salad person, but there are times where I've enjoyed a salad really well. Usually with lots of meat, or the hardest thing for me is I like blue cheese dressing. It's like no ranch, let's just blue cheese.
I'm a vinaigrette.
Yeah, vinaigrette. Yeah. Yep. I've got a few friends who are vegetarian. Of course, they're all old people like me, so they post all their food on social media. And I was like, okay, that salad actually looks good. Yeah. All right, but anyway, so we've talked about making our nucleotides. We can also catabolize them as well too, break them down. So we're putting in the same chapter, unlike our fatty acid oxidation and our fatty acid synthesis. Here we're cramming both of these at the end of this chapter here. So what happens if it's time to recycle our purines or our pyrimidines? We'll start with our purines, AMP and GMP. They both start with this breakdown where we have, we're going to remove our base. We're going to remove our phosphate first, and we remove the base. So we're just left with the, or I guess the sugar. So we're just left with the base there. Say with adenosine, kind of in the process, adenosine gets converted to inosine and then hypoxanthine. Both of them will ultimately get into the form of xanthine in here. So this is a keto form, and then we can convert this into uric acid. Okay, and this requires xanthine oxidase. It's really kind of interesting because spiders and other arachnids lack xanthine oxidase. Fun fact time in there. I'm not going to ask you guys about that. There is an interesting aspect of this though, because let's move this over here. Let's do this down here. Yeah, that'll work. So our uric acid, we can convert that into allotonin, allantoate, or urea, so on and so forth in there. And that just kind of depends on the organisms. Our primates, we kind of leave it at uric acid, but most mammals will further make allantoin. Bony fishes, go to allantoate, and then we get the urea for our amphibians and so on, and then our marine animals can convert it into that. So multiple stages in there. You've kind of sort of seen that before, but this is again breaking down our pyrimidines. Okay. Oops, come on. Maybe it was mentioned because, but it's not something because we stop here. I don't emphasize this. Yeah. But that's just something. If you were to go like so our primates, if you go poke through primate poo, go to the zoo and collect some gorilla poo, you're going to find lots of uric acid. But if walking your dog or find deer poop in your yard, like I do pretty much every day, the deer love my yard, then we'd see more allantonin in there. Right. I know. Yeah. And I'm on like the deer highway between. All right. But there is one thing that happens, our uric acid. All right. So we normally excrete that, but excess uric acid can lead to gout. And uric acid often accumulates in the joints, particularly the extremities, toes and fingers. And what happens is the uric acid becomes sodium urate, and it will crystallize. So you actually get solid little crystals in there, and it's really painful. Do I have the picture? I do not have the picture. Well, it is I can't say medieval, a couple hundred years ago, somebody was illustrating gout who suffered from it, and they had a picture of like this little devil stabbing, someone's toes, because apparently it is incredibly painful because you're getting solid crystals in your joints. So yeah. Yeah. And, I say it primarily affects males, but I know lots of women who have it. This may deal in part with underexcretion of urate. Also, if it is diet dependent. So if you consume a lot of alcohol, eat a lot of fat, it used to be known as, the rich man's disease because, in medieval times, people who ate a lot of fat and drank a lot of, had the money to buy these expensive, rich foods, would consume so much, they'd make a lot more uric acid, and then that would lead to the gout. Yep. Yeah, that's true because depending, and it depends a bit on the diet there too. But avoid purine-rich food, so don't eat a lot of liver, don't eat a lot of seafood, and avoid fructose is one way to kind of treat it. There are particular medications out there like allopurinol, which is a xanthine oxidase inhibitor. So if we go back, here's our xanthine oxidase. Convert that to uric acid. Well, if we inhibit this, we're going to leave this in the xanthine form and not make as much uric acid. So we can slow down the production of uric acid. So what did I want to say about that? Yeah, there we go. So allopurinol will bind to xanthine oxidase and get converted to oxypurinol. So we're kind of slowing this down. All right. There is another method to treat that. So allopurinol has there are various pharmaceuticals that have that, but there is a old wives' tale or traditional medicine. If you have gout, eat lots of cherries or drink cherry juice. Any ideas why? cherries are naturally high in allopurinol. So kind of getting the same deal there too. Kind of like, you know, if in the old days, if you had a headache, they'd say chew on some willow bark or make willow bark tea. Well, that's a salicylic acid, which is the precursor to aspirin. Yes, yeah. I'll find it and post it because yeah, there's a nice little demon there stabbing somebody in the foot because it's so painful. All right. But yeah, so understanding these pathways and figuring out, you know, we want to, you know, catabolize these, but maybe we don't want to make a whole bunch of uric acid all at once. Okay. All right. If we look at pyrimidines, we have a slightly different pathway. Okay. We're going to end up making this methyl malonyl semialdehyde at some point. Okay. And again, here, instead of stopping at uric acid, our pyrimidines are being converted to urea and ammonia. Okay. And we can also get some intermediates of the citric acid cycle. So you can see, alpha ketoglutarate, succinyl CoA, thymine gets degraded to succinyl CoA, but you get this weird methyl malonyl semialdehyde. Eventually, though, you're going to get to urea. We're not going to focus on that pathway in there. I like gout. I've almost always asked questions about gout. Sometimes, more recent years, maybe not. But the key is, if you see gout, you should start thinking, oh my, purine, purine? Yeah, purine salvage pathway, or purine catabolism. So we can catabolize them into uric acid and urea, but a lot of times we can recycle them from pathways in here. So we take our, phospho-ribose with the nucleobase on there. Well, as we start to degrade them, we can release the free bases, and we can free base. Hey, Richard Pryor. Okay. Richard Pryor. Yeah, that's probably before your time. Free basing.
Oh yeah, I know.
Yeah. Okay. Yeah. When he's free basing cocaine and badly burned. Anywho, anything that sticks in the memory. Okay. So yeah. I'm waiting for Will Smith to come in and slap me because I'm being disrespectful with my jokes. But yeah. It's like, yeah, like Chris Rock. I got jokes. Okay. But the free bases can be reused. And you'll see this adenosine phosphoribosyl transferase. We can just stick them on another ribose in here. The brain especially uses these salvage pathways. Okay. If we have, and of course, because there are so many enzymes involved in all these pathways, if you see something mess up, like if your xanthine oxidase is inhibited, you're going to see gout. This hypoxanthine guanine phosphoribosyl transferase, if that's deficient, you lead to what is a nightmare disease. And when I first read about this is the one that makes me cringe, Lehninger syndrome. So that's a deficiency of this particular enzyme in here in this salvage pathway. And it's early onset, so you'll see this in children. There's neurological impairment, but they exhibit a particular phenotypic activity of finger and toe biting. They will literally chew their fingers and toes off. And I'm not sure why that happens, but it's results from this lack of this enzyme. Lehninger syndrome is just, yeah. Yes, yeah. So it's neurological impairment because you have a lack, maybe it's a lack of purines in the brain or something like that. But it leads to, you can't get the salvage pathway. And keep in mind, at the early stages, we said nucleotides are kept low. So if we can't incorporate these in here, if we're missing some, we can't salvage these, then you're going to see neurological impairment. Most primarily because the brain is especially dependent on these salvage pathways. We're not making a lot of nucleotides up there, so whatever neurological impairment happens results in this behavior as well too. And whew, it's just nasty. It is a scary. And every time like our kids, this is the terrible thing about being a biochemistry professor. Our kids suck their thumb when they're babies. I'm like, just sucking, right? Not biting. Yeah. Yeah. So it's and it's really rare. So well, it's a rare disease, but not something that we typically worry about. And again, just to go back to chemotherapy, talking about cancer, a lot of cancer drugs are, analogs. They target nucleotide biosynthesis because, again, fast dividing cells, we want to prevent them in here. So you might have glutamine analogs, which will inhibit those glutamine aminotransferases, slow that down. Fluorouracil, it's actually really fascinating because this will go through the pathway and it will make the uridyl, the deoxyuridyl, but incorporates this fluorine in there. So you can't actually then convert the uridine to thymidine. That fluorine atom in there is in the place where you would add the methyl group, and that the presence of that fluorine inhibits the chemistry that would make that, so you can't make thymidylate. Methotrexate and aminopterin will inhibit dihydrofolate reductase. These are our traditional competitive inhibitors. They just bind in the same site and slow stuff down, right? What has been interesting is, much like AIDS medications, where you have 3 different approaches, chemotherapy, you can often find a multi-drug regimen to be better cooperative than a single drug in there. And you just kind of tailor it to the patient. What can the patient handle? Okay. Many antibiotics also target nucleotide biosynthesis. So allopurinol has been studied against trypanosomiasis. Trypanosomes, I was always a little nervous in my lab in grad school because I had my particular project with the cytochrome BC1 complex, but one of the other guys in there was studying trypanosomes. My advisor had connections in Kenya and was looking at African sleeping sickness, and that's transmitted by, I believe it's mosquitoes, some type of the trypanosomes live in there, and there's a whole life cycle where the cattle will be the host, then the mosquito will feed on them, and then bite the humans, and then so on and so forth in there. I lived on a cattle farm. I'm like, oh, I want to make sure I'm really clean, so I don't bring home any trypanosomes when I go visit the family there. But again, trypanosomes, have to acquire nucleotides from diet and salvage. They cannot make them de novo. So trimethoprim, again, another antibiotic that inhibits the bacterial dihydrofolate reductase, but it binds to the bacterial version much better than the human version. So you might see like a slow, a very mild decrease in the human form, but the bacteria, so these prim type drugs are antibiotics that are going to be targeting their nucleotide biosynthesis. Okay. All right. So that's a lot in this chapter. All right. The big thing is, how do we make amino acids? How do we make our nucleotides? And then how do we break down or salvage our nucleotides? All right. Like I say, this next exam, I think, is going to be entirely take home. So the biggest thing to study is to just understand the pathways, understand what's going on, and then being able to kind of apply that outside of standard question. I'm not going to make you guys memorize a bunch of stuff. Questions about this chapter? I was going to say the sad thing is, oops, that's the wrong one. Close that one. Yep, we got to start another chapter. We got time, 30 minutes. So, yep. And the good news is it relates. Yeah. So 22, 20, 21, 22, and 23. Yeah. So I think as tough as chapter 22 is, because it's just so much stuff in there, I think you'll like this chapter a little better. Yes. This is all semester we're leading to this chapter. All right. And then after this chapter, we're going to like do something different. No more metabolism. So like, yay. So we just got to get through this last chapter. 119 slides. So it's long, but it's not as, I don't think it's as horrible, because now instead of, well, we're looking at this pathway, well, we're looking at this pathway, now we're looking at everything integrated. Now, some people that might be worse, but we can actually we're basically going to be looking at the whole organism and not just a particular section of an organism. So we'll start off today covering the basics of endocrine signaling, and then on Thursday we'll see how much we can talk about this. But we're going to look at insulin, and diabetes and obesity. So that gets a little more fun because we can look at stuff and we can talk about various diets, the whole low carb stuff there. But let's see if we can get through the introductory part here as well. So everything we've looked at previously has been at the cellular level, and we may see, okay, well, the liver does this, and then it sends it in the bloodstream to the muscle where this happens, and then it goes back, and so on and so forth in there. So we've looked at specific enzymes, specific pathways, specific regulation of particular pathways, and then how we transport stuff. That awesome question about you've got this size fat. Small fatty acid will go directly across the membrane. Medium sized fat needs a particular, like, was it the acylcarnitine transporter, and a large fat needs another transporter. Right. With this chapter, we're going to look at the whole thing. So we're going to kind of talk about the roles of tissues and organs. We're going to talk about flux of metabolites between organs in there, and hormonal regulation, because our hormones, they can be local, but a lot of times an organ will release a hormone, and we'll see that travel throughout the body to other organs and have effects there. And we'll talk about control of body mass. It's like, which doctor you just ready to talk about this year? Because I'm controlling my body mass. All right. Woo. Yeah, I'm gonna be Richard Simmons next. Okay. That's an old reference thing. Okay. So neuronal signaling is where we have our neurotransmitters, and we can release those, and they can act on a nearby cell. So these are very short distance. We're talking like micron level distance here. Our nerve impulse travels, even though our nerves run through our whole body, it's basically one nerve cell releases something, talks to the next one, talks to the next one, so on and so forth here. So our distance is typically very small. Hormonal signaling, we're talking about 1 meter, 2 meters, and maybe find somebody who's like Manute Bol, or who's the other really tall guy in the NBA right now, 7 foot something. Not Giannis, yeah. But there's I can't remember. There's some other guy. Maybe it's I can't remember, but I think he played for Baylor in college. But yeah, it's really, really tall guy. There's very few people over 7 feet.
We just gonna go with Shaq.
Yeah. But we maybe two meters or more here. So we're looking at hormonal signaling very far away. This is not a cellular level. They're going to be targeting things. They're going to go through the bloodstream usually and target something someplace else. Now, some hormones, it's funny, they get released by the cell and then bind to extracellular receptors of the same cell and come back and affect it. Was an autocrine, but others will go farther away. Mm-hmm.
How is it 1 meter of distance?
Yeah, just saying, like, maybe your pituitary gland releases something and it has an effect on your pancreas, or your pancreas. Yeah, here's a good one: adipocytes. Your adipocytes release a hormone that will travel to your brain and say, "I'm hungry, feed me, Seymour." Okay. Yeah. So that's why we're getting that kind of 1 meter thing there. Okay. All right. So the issue with hormones is goes a long way. Hormones are produced in really, really small amounts. And I always like this, if you wanted to look at thyrotropin releasing hormone, you basically had to cut up 1 million pigs, because you needed 1 million hypothalamuses to purify enough TRH to study it, which is fine because, people love bacon. So we can save the hypothalamus for me, right? All right. But yeah, I always think of that the same way. If you wanted like 1 mole of Titan, this really huge protein, you'd have to chop up a lot of people to get that. So not going to happen. Now, because hormones are so low concentration, this is why radioimmunoassays were developed and are still used in some of these studies because they are the most sensitive. Like most of the stuff you'll work with, you'll probably use like horseradish peroxidase or was it ammonium perphosphate? Is that right? Ammonium phosphate as detection because you can tag those antibodies and detect them, fairly small quantities in there. But hormones, sometimes that's just not going to work. So you can radio label them and you can detect very small amounts of radiation. We're really good at detecting radiation. So that still happens in there. How do we do this? You can take a purified hormone, inject it into an animal, and the animal will make an antibody to that hormone. Okay. So this is why if you've ever used something like mouse hormone or anti-mouse something tags in there, you can see okay, we made this in mouse. We injected something into a mouse, and we got this mouse version of the antibody to it. Antibody is purified and labeled with a radioactive tag or an enzyme that produces a culture product, depending on whether we're doing radioimmunization or ELISA. For a quantitative assay, we add a known amount of tagged antibody to the sample in here. And then the fraction of the antibody gets measured, that gets detected, whether by photometry or radiation, it tells us where that antibody is and what it's doing. The neat thing, hormone receptor interactions are really specific and very, very high affinity. So that's one of the reasons it only takes a small amount of hormone is because they're going to bind to their receptors really, really well and trigger whatever effects they have there. So different types of cells, different types of receptors. So, like thyroid stimulating hormone might bind to, some cells because they have the right receptors, but it's not going to bind to muscle cells because they don't have the correct receptors in there. Okay. We can have isoforms of the receptors as well too that will affect structurally similar hormones can often, even if they're very, very similar, they can bind to different receptors because they are so specific. The interactions are very high affinity, so only low amounts are needed. Okay. Again, what we're going to see is when hormones bind, we have a signal cascade. So just like we've seen before, it binds to receptor and triggers downstream effects. So usually there's a secondary messenger like cyclic AMP or a inositol triphosphate. Because I'm 8, it's like IP3 leak. And these will allosterically regulate enzymes inside the cells in here. We might see a tyrosine kinase, so we get a phosphorylation cascade, or we'll see a hormone-gated ion channel, just like we saw, was it voltage-gated ion channels? You change the potential across a cell and a channel will open or close. Here we have a hormone-gated one where it binds to a particular spot and opens it or closes it.
Is this all connected to like also the
Yeah. And those may, and that may be more for this last one. Our steroid hormones bind to a receptor protein in nucleus and alters gene expression. So that's certainly going to be affecting puberty as well too. But we can also have our adhesion receptors that affect the cytoskeleton too. So, and this is just almost kind of a review of our signal transfer from chapter 11 or 12. I can't remember which one exactly there too. Okay. Hormones can be extracellular, which is what we're going to talk about most, but they can also be intracellular. They can be released, and immediately bind back to the same cell and affect it, and affect the same cell there. Where they can be released and go to the nucleus in there. And that's true for even if they're released far away, they can bind to the cell surface, or they can get taken into an endocytase into the cell and go to the nucleus. Go directly to the nucleus. Do not pass go. Do not collect 200 phospholipids and bind to various transcription factors in there, which it's stunning to me how compact we're going to make that, but it has to be that way. Yeah. Peptide and amine-derived hormones typically bind extracellularly, Insulin is a peptide hormone. Epinephrine is an amine hormone. They bind to the receptors that span the membrane and introduce a conformational change that gives us a secondary messenger. So if you think of like our G protein coupled receptors, our GPCRs, those would act that way and give us signal amplification in lots of targets. All right. For mammals, we got three main classes. Paracrine hormones are released into the extracellular space and they diffuse nearby. So an example is eicosanoids. So an example would be my awesome leg, which is now just mostly kind of yellow, a little bit of red down there. I've certainly released some hormones in there that dealt with wound and wound healing, and they just diffuse into that area. So they go outside the cell, but they don't go very far away. They just stimulate the cells nearby. Endocrine are released into the bloodstream and carried wherever the hell they need to go. So this would be insulin and glucagon, good examples there, and we'll talk about those a lot. Autocrine are a little bit different. They affect the same cell. They get released by the cell, but then they immediately bind to receptors on the surface of that cell and trigger changes within that same cell. All right, so that sounds a little weird, but autocrine is a third class in there. So autocrine would be affecting the same cell, paracrine, nearby cells, endocrine, far away cells, different organs, and so on. Okay. That would actually work alphabetically, AEP. Okay. Or maybe you do reverse alpha. Anyway, these are different types of hormones. We can classify them. You're not going to need to memorize these: eicosanoids, steroids, vitamins, retinoids, thyroids, so on and so forth. And it just kind of gives you a general idea of how they're going to affect things. A lot of our peptide or eicosanoids are plasma membrane receptors, second messengers. A lot of these retinoids and thyroids are nuclear receptors, and they'll affect transcriptional regulation. And if you look at nitric oxide, it's a weird one because it's got a cytosolic receptor and a second messenger. So it's similar to the first class, but because it's a gas, it just kind of diffuses into that across the membrane. It doesn't bind to a membrane receptor. It diffuses across the plasma membrane and binds to a cytosolic receptor. And then from the inside triggers a second messenger. Insulin is a peptide hormone. All right, so I don't know how far we'll get with insulin, but we're going to spend the next long section talking about insulin. Probably. It's the one we talk about the most. And one of the reasons we talk about the most is because it's so well studied. We know a lot about insulin and insulin receptors, and we know a lot about disease states that arise like diabetes and so on and so forth. But there are others out there like glucagon, very similar to that, somatostatin, and so on, ones that we know less about and that are less common. So insulin is synthesized on the pancreatic beta cells as a pre-pro drug, pre-pro insulin. So it's got an inactive form, an inactive form. So two things need to happen in there, but it gets processed into this very small active form and then stored in secretory vesicles in our pancreatic beta cells. Okay. So here's what we make. Our pre-pro insulin is this long single unit in here. We have our signal sequence that tells it where to go. From the ribosomes to the secretory vesicles in there. And we can see we have a couple cysteines in there. Once it gets to its proper place, first thing we cleave off the signal sequence. We don't need it anymore. That just tells it where to go. So now we, instead of having pre-pro insulin, we have pro-insulin. Okay. And our pro-insulin, besides cleaving off the signal sequence, you see we make these 3 disulfide bonds. Okay. There we go. All right. Our proinsulin is then acted on by another enzyme, which cuts out about half of it, which is known as the C-peptide. All right, so we get rid of the C-peptide, and now we have this A chain and B chain that are connected by a pair of disulfide bonds. And this is our mature insulin that will actually be capable of binding to receptors and triggering effects in the cell. Okay. So pre-pro, we have our reduced state and our signal sequence on there. Our proinsulin, it's oxidized, but we still have the whole signal, the whole chain except for the signal sequence. And then mature insulin, we lose the C-peptide. Okay. All right. So insulin is secreted in response to blood glucose levels. Eat a meal, start producing glucose in the bloodstream, and insulin is going to be released and say, hey, here comes the insulin, or here comes the glucose, let's do stuff with it. So it'll bind to receptors in the muscle, the brain, the liver, the adipose, and basically any organ that metabolizes fuel. Okay, so in muscle, insulin will synthesize glucose uptake. You've seen those little gels, long distance running or biking or swimming or whatever, you have these little gel packs, and you can get those gels in there. They have a lot of sugar in there so that your muscles will take up glucose, so have the energy in there, so you don't presumably don't hit the wall. In the liver, it promotes glycogen synthesis because now we got excess glucose. Let's convert it into glycogen, so we'll have energy for later, our medium-term energy. So muscle is immediate energy. Liver is medium-term energy, and adipocytes, we're going to promote glycerol synthesis and inhibit the breakdown of fats. So this will be long-term energy storage. Okay. Okay. So that's just kind of a very quick overview of insulin. There are other similar peptide prohormones that can be broken down into multiple other drugs. So this is a convenient way for the body to be ready to trigger a whole range of effects. So this pro-opiomelanocortin, or POMC, as I'm going to call it from now on, which always makes me think of the pomegranate. It's like Palm Wonderful at the grocery store. Today's lecture is sponsored by pomegranate juice now. But this POMC has several different cleavage sites. So just like we saw insulin, you cleave out the C peptide to make the active form. Our POMC can be in this pro form, and then depending on where you cleave it, you can get 10 different peptide hormones out of it, like beta endorphin, melanocyte stimulating hormones. That should be an E in there. Mutations in this gene are associated with obesity too. So let's kind of generically look at this. Here's our DNA. Here's our mRNA. Our mRNA gets converted into our pre-pro drug in here. We have our signal peptide. And then once we start processing our pro-POMC, you can get our melanocyte stimulating hormone, or ACTH, lipotropin. You can tell by the names of these, lipotropin and melanocyte, that these are various. These can be associated with metabolism of fats in there. But you can cleave out this little tiny gamma MSH. You can make this ACTH, which can be further cleaved into alpha MSH. And you can get this longer version, beta lipotropin, which can be cleaved into gamma lipotropin and beta endorphin. And then you can further cleave that into beta MSH and so on. So you get, what is this, 6, 7, 8, 9 different compounds. Well, I think beta lipotropin is not active. I think only these are active. So 8 active forms in here. Okay. And the whole key is, you don't need to make very much. They're very, very specific. And then you can have this pro form ready to convert into what you need in small quantities to see the effects in there. If we look at things like epinephrine and norepinephrine, these are catecholamine hormones, or amine-derived hormones. And these in particular are synthesized in the adrenal glands. So here we have epinephrine. You can see the base molecule epinephrine and norepinephrine. Very similar. We just have a methyl group over here. They are synthesized from tyrosine and held in storage vesicles, much like our peptide hormones, and then released as needed. Yes.
Issue.
Uh-huh.
And I got the issue too. You put yesterday for chapter 23.
Yeah. Yeah. Yep. Yeah. So it should be fixed now. It should say like next Tuesday, I think April 5th. For
22.
Okay, maybe 23 didn't save, but I'll fix that. Yeah. So yeah. Don't sweat it. Those are guidelines by when I'd like you to have them done, but they stay open.
Okay.
Yeah. No worries. Yeah. But again, our catecholamine hormones are very similar to our peptide hormones in that they are held in secretory vesicles and released as needed in there. Now, on the other hand, if you smack yourself in the leg with a tree, your eicosanoid hormones are not synthesized in advance. They are made as needed. and those depend on, arachidonic acid, which you have to get from your diet, and various phospholipases. Phospholipase A2 will start cleaving these and making the drugs in here. Paracrine hormones, which are going to act nearby, and they play a role in inflammation, smooth muscle contraction, platelet function, all that good stuff in there. So they're made as needed, typically in response to injury. All right. Steroid hormones are made from cholesterol. And these will include things like cortisol, testosterone, estradiol, all that good stuff. They will bind to carrier proteins because they tend to be rather hydrophobic. So they need carrier proteins to get through the bloodstream. And then they can bind to their receptors. They can get endocytase into the cell, go to the nucleus, and bind to their receptors in the nucleus to alter gene transcription. Some look like they can also bind to a plasma receptor. I know cortisol is involved in pain response and inflammation, so that may be involved with a plasma receptor in here. And you can see some other variations of these as well too. Vitamin D also binds to a nuclear receptor. And last semester, did we talked about vitamin D deficiency and something? Do you guys remember melanin?
Yeah.
Calcium? Yeah. Okay. All right. Yeah. So, but vitamin D binds to a nuclear receptor, and we get it from, the sunlight or from diet. Something like 90% of the world's population has a vitamin D deficiency. Many not extreme, but a lot of people could use some more time in the sunlight, or this is also why you see milk or vitamin D added to milk, because it also helps in calcium uptake as well. So you see calcitrol in there. Calcitrol is the active form of our vitamin D hormone, and it affects transcription of genes that regulate calcium balance in there, and does some other stuff as well too. But retinoid hormones will also go and bind to the nuclear receptor. We get them from vitamin A. So you notice this A, D, I don't think we talked about vitamin K, but E, all of our fat soluble vitamins are ones that seem to also have some hormone effects in here. All cells have at least one form of this retinoid receptor. So I think we mentioned last week the RXR retinoid X receptor. There's like an LXR as well too, but all cells have some type of retinoid receptor in there. And this hormone receptor complex is important in regulating cell growth and cell differentiation. So that cell needs to grow to a certain size. When it gets there, it can divide, differentiate. That's why the retinoid receptor is so important for all cells. And you notice that they all have at least one form of the retinoid receptor. Some will have multiple forms in there. These retinoid receptors are most active in cells that are experiencing rapid growth. So you look at epithelia, skin, immune system, cornea. Exactly. Yeah. So you start to lose plasticity. Part of that, there's a variety of things that combine with that, but these RXR receptors and slowing that down in there as well too. It's why maybe vision starts to go when you get old as well too, and all that good stuff in there. Thyroid hormones bind nuclear receptors. So I like this, the T3 and T4, which are conveniently named because T3 has 3 iodines and T4 has 4 iodines in there. All right. And it's a precursor to this thyroglobulin, which can yield T4, which can be converted to T3. And when it binds to its receptor, it helps regulate expression of enzymes that yield energy. Okay. This is why if you have a hyperactive thyroid, you may have a lot of nervous energy, and people with hypothyroidism tend to be thinner and maybe fidgeting around and stuff like that. People who have hypoactive thyroids tend to have less energy and are prone to gaining weight as well too, because they're not. I'm not sure. I've looked into the thyroid a bit, and it's one thing I learned is something like 90% of middle-aged adults will have some anomalies, nodules that will grow on their thyroid, and I'm stunned to learn so many people I know have thyroid issues. I would say most people I know have hypothyroidism, where the thyroid is just not as active, but I'm not sure which one's associated with cancer. I do know that thyroid cancer is relatively rare. Also, quick quiz for Duck.
You said it's associated, but it doesn't decrease your chance.
Okay.
Yeah, it doesn't like increase or decrease your chance. Both people have.
Okay. Okay. So anybody concerned about nuclear war?
Yeah, a little bit.
Yeah.
I live in Europe.
Yeah. It's interesting because a friend of mine that I went to college with is like obsessed with the Ukraine war and Russia, and Russia's a bad man, yeah. And he's like, is anybody else worried about nuclear war? So I was telling him, I was like, dude, just think back to when you were a teenager, and like when we were teenagers, there was a constant threat of nuclear war and the Cold War, and Berlin was a huge party city because if there ever was a third world war, Berlin was going to just be the first place destroyed because it was in the Soviet held areas. It was fascinating to see the way the culture was and then how it changed over time. So I was just teasing him and he was laughing about that too. It's like, okay, that's true. Yeah. But there are people who are worried about nuclear weapons being used. And that has manifested in iodine tablets selling like crazy. Why iodine tablets?
Because they can fight with radicals or
Specifically, nuclear weapons will lead to radioactive iodine in the atmosphere. Radioactive iodine gets incorporated in the thyroid for these T3 and T4. So that's where you most likely find iodine in there. So if you have radioactive iodine in your thyroid, you're going to see radioactive damage, and that's thyroid cancer or damage or something like that. That is a big risk there. So iodine pills are, of course, non-radioactive iodine. You take the iodine pills, it outcompetes the radioactive iodine so that you don't accumulate radioactive iodine in the thyroid.
that makes sense.
Yeah, yep. And it's so funny because now there are companies who are like, yeah, we used to sell like, whatever quantity we would sell in like 6 months or a year, we've sold in a week because enough people are worried. I'm not terribly worried, but that's out of the scope. Yeah. And people say I would suspect honestly the most likely thing they would do would be a tactical nuclear weapon, which is a small scale. But the problem with that is the winds blow from west to east. So if Russia nukes Ukraine, the fallout is going to be in Russia. Of course, the way Russia is. Yeah. It's like, and they take all their poor conscripts from the poor areas of former Soviet Union states and throw them in there with minimal training and crappy equipment. Oh, it's an indictment of the Soviet socialist system and philosophy of government. Bad. Anyway, nitric oxide. We don't think of that as a hormone because it's a gas, but nitric oxide is a nice little hormone. It is essentially a free radical made from arginine and oxygen by this nitric oxide synthase. There is inos and enos for endogenous and inducible nitric oxide synthase and endogenous nitric oxide synthase. It acts near its point of release because it just diffuses as a gas. It can diffuse across membranes. So it enters the target cell and binds to a cytosolic receptor. And this leads to this ultimately regulation of blood pressure, lowers blood pressure in there. So inducible inos is a good target for drugs in there. All right, but I could talk about nitric oxide synthase for a very, very long time, but apparently I just have it on one slide. So we're not even going to worry about inos versus enos. If we look at our major endocrine glands, so we're almost at the end of today. What slide are we on? We'll see here in a minute. Our major endocrine glands include the hypothalamus and the pituitary gland in the brain, our thyroid, which is why it's so important. It's like people don't think about their thyroid until it goes bad or something like that. We've got our adrenal glands behind our kidney, our pancreas down here in our gut, and then adipose tissue. That's one we don't think about, but we're going to see on Thursday, adipose tissue is important in releasing some appetite and appetite related hormones in there. And then of course hormones for ovaries and testes for men or women. We're getting into crazy biology stuff now, right? I don't know. Maybe I'm qualified to be a Supreme Court justice or to lead the NCAA. Anyway, yeah, politics and crap.
Yeah, that interview, some of the interview questions.
Yeah. There's so much. You see so much. Yeah, I'm gonna get you. Yeah. So it's just dumb. All right. But wrapping up today, the reason that we have all these different tissues, all these different organs, is its division of labor. If you go back to the simplest organisms, they're single-celled organisms. One organism has to do everything. An analogy I like is society. You start off as like individual small groups. Maybe I gotta be able to do everything. Towns, you find like somebody could be a blacksmith, somebody could be a good hunter, somebody could be a good gardener, somebody could make good clothing, somebody could build shelters, and you start to get specialization. And so now, and I think back to early computers, we kind of had to troubleshoot a lot of stuff. But now interfaces, one of the complaints that we hear from older people in faculty is like, kids today are supposed to know how computers work. It's like, no, everything is so optimized now, it just works. And if it breaks, maybe you can YouTube it or you have somebody else repair it, but it's a lot harder to get under the hood and see. You go back 20 years, you had to do a lot of troubleshooting. And I still am stunned that I've coded one line of code in my life, and I'm stunned that I figured out how to do it. But just like, all the specialization that we have in society, we have specialized functions for our organs, our small intestine, portal vein, liver, all the stuff that they do. They are specialized to store energy or to, take in everything or to, regulate everything in there. I used to joke, well, I joke now that I used to think the brain was the most important organ because it kind of controls everything. It's our seat of consciousness, it's where we think. But really, it's probably the liver. Because, you can live without a brain, see politicians, but you can't live without a liver. It's got live in its name, right? Okay, terrible. All right. So where we'll end today is our hormones and our target tissues. We get input from the environment. We'll say sensory input, but that can include a lot of different things in here. Whether this starts at the central nervous system or starts someplace else. Something happens here, goes to our hypothalamus, and the hypothalamus says, okay, I got to react to this, so I'm going to release some hormones. And then these can affect the pituitary glands. So we see our origin of signals, our first targets, our second targets, and our ultimate targets, which can be multiple steps away. But the anterior versus posterior pituitary will release hormones that can target different classes in there. We might see the adrenal glands get activated, or the thyroid, or the ovaries, or we might see, in terms of like a diet, the pancreas, our medulla oblongata. But ultimately in here, many tissues, muscle, liver, so on. But we have this cascade effect, just like we saw our signal cascade within a cell, within the organism. We have our initial reaction, and then other stuff. It's like, oh, this is this type of environmental stimulus. I'm going to trigger these things to deal with that, hopefully. All right. So next week, geez, it's only Tuesday. On Thursday, we will talk more about hormone signaling, and I think we will probably get into insulin regulation and dietary regulation and all sorts of stuff like that as well too. All right, but that's where we are. Woo. Good stuff, right? Yeah, it is a lot.